Good morning.
Good morning, sir.
After a time gap, I'm again starting the topics, some of the topics, and mostly I'll be covering the general and systematic animal virology. We'll try to share informations about the viruses and try to take some concept about the viruses, their nature. Then we'll discuss about important viral diseases that affect the animals. Probably most of the viral diseases, as I could see, you have already studied in pathology and some other courses, but still in microbiology point of view, we'll discuss them. We'll start with the basic concept about the viruses. As why we are here, it is because of the viral pandemic. So we'll try to know about this, the different types of viruses and the viral infection. But before that, one small topic which has been left out in the biotechnology part is about bioinformatics. So in this particular session, we will cover this bioinformatics, the concept of the bioinformatics. It's a very brief outlines only. We'll discuss it. And in the afternoon, again, we had a class from 2 onwards, a combined class. From that point onward, I will start the virology. So hopefully everything is going fine with you. Can you see the slide?
Yes, sir.
Is it okay, this appearance of this slide? Or can you see the slide now?
Yes, sir.
Fine. So later on, I'll provide a small notes on this aspect of bioinformatics, and it become a new field and new subject. Now has been incorporated in this course, microbiology, particularly animal biotechnology part. But this is not something is restricting to the microorganisms. Rather, in all biological field, nowadays people use this particular science of bioinformatics. And basically, this has, again, I should say, revolutionized the process of learning, particularly interpretation of biological data. So this is one subject area where there is integration of biology with information technology, where we apply computer-assisted statistical models for interpretation of biological information. So this is really a very, very fascinating field of biology emerging these days, and where we use some computer software for interpretation of biological data. Just to give you a very preliminary and brief idea as related to this present situation, you have heard about that some UK strain of SARS-2 is circulating in India. In India, we have Brazilian strain. We had a South African strain. Now, how people could able to tell that this is an UK strain, this is an Brazilian strain, or this is something like a double mutant? These are some of the words that on daily basis you are learning from these medias. Now, this is very well possible by using some. Yes.
Sir, excuse me, sir. Some students are waiting.
Okay. Yeah. when we say that a particular strain of SARS-2 COVID-19 strain, so this is very well possible by using some computer-assisted software to analyze the biological data in the form of the genome sequence, the nucleotide sequence of the viruses. As you know, the virus started in 2019 only. It's a new kind of virus, but now we could able to track out this particular virus is related to what other type of viruses prevailing in this globe. And for that, to generate that kind of data and information within a very short span of time, it is the use of bioinformatics tools. If anybody learned this, how to use this software and have a basic idea about the nucleotide sequences of the virus, even you can also use this software and can go for some sort of analysis and can understand it. So I'll be focusing on this particular new area, bioinformatics, but it's not possible to have a complete whole on this subject within a 1 hour class, but try to understand about the concept of bioinformatics. So in context to SARS-CoV-2, SARS-2 virus, we will give you some highlights and some informations, and how best in our institute we have used this science of bioinformatics in 2013 to establish the epidemiology of PRRS virus that hit the state of Mizoram in 2013, where from it has disseminated and how the boundary transmission takes place. So this information with scientific data we could able to generate by using the science of bioinformatics. So in '87, Dr. Wallim first he given the concept about this new area of bioinformatics. And it was a very preliminary concept, but was not properly understandable to everyone. He says that bioinformatics is a new subject of genetic data collection, analysis, and dissemination to the research community. This is a quite interesting area. As it is very recently, the science has been developed. So one of the very fascinating part of this subject is that the data are open access to all of us. Anybody can access this biological information stored in certain supercomputers, and it is open access. Anybody can access, and if you wish, you can download the entire genome sequence of any kind of viruses, particularly the SARS-2 virus. If you want to download the entire genome, any one of you, I will show you how to do it by simple using the Google search engine. So it is a science which also related to dissemination to the research community, means this informations can be provided to every one of us, not restricting to a group of people. So further, people put more highlights and says the definitions as an integration of computer, mathematical, and statistical method to manage and analyze biological information. As such, the biological informations are a very huge type of information which without any kind of computers, people cannot handle and manage it. That is why this is an integral part that we must use some high-end computer programs and mathematical and statistical methods to analyze the biological information. As I said again, sitting in your room itself by downloading the different genomic sequence from the open access areas in the internet, you can analyze how much these viruses are related to each other, or not only the viruses, even two species of animal, how much they are related, two species of bacteria, how much they are related to each other. These things can be analyzed with high accuracy by using the tools of bioinformatics. So for quite a long period, people were not having much information about the organisms, means their the evolution process. In the evolutionary process, which are the organisms which are more closely related, some are distantly related. This kind of analysis were limited earlier. But once this particular field has developed, and now it become very easy and very accurately we can compare and interpret the organisms, their relatedness. So all aspect. Even probably you have heard about in some of the scientific journal, people are exploring the evolutions of humankind and the possibility of like how the human migration takes place, all on the basis of these bioinformatics tools, and the harboring some special genetic materials in our ancestors. Even from fossilized samples also, people are able to get the information, and they are trying to give some interpretation of those fossilized samples. So this is really a very fascinating and very fast interpretation we can make it, and that too also with high accuracy. The way the people could able to make a vaccines, and now we are able to take some recombinant vaccines, it is only possible because of the development of bioinformatics tool. Probably it was not there earlier, that's why it took a long time to develop some good quality vaccines and drugs, likewise for our use. Then again, another person, he says the field of science in which biology, computer science, and information technology merge into a single discipline. This is a more appropriate kind of and very simple way we can explain it. The bioinformatics is the field of science in which biology, computer science, and information technology merge into a single discipline. These are integral part. Simply generating some biological data in your laboratory, it is not possible to go for interpretations without use of information technology and computer science. So this bioinformatics, we can say it's a field of science in which biology, computer science, and information technology merge into a single discipline. Okay. And I will say that this is not restricting to the microbiology. In all sphere of biology, people are extensively using this science and interpreting the biological information, and we could get some valuable data out of that. And this particular process has help us to design some drugs, new drugs, to design some vaccines. And very easily we can analyze the epidemiology of disease. What are the different strains are circulating in an area? To identify and to analyze these results, we use the bioinformatics tools extensively. So again, the same way in 2001, Lascombe, he explained the bioinformatics is conceptualizing biology in terms of macromolecule and then applying informatics. First, we need to generate the data by wet lab experiment. That means in our laboratory, by doing some biological experiment like by polymerase chain reactions, by extracting the DNA, by extracting the RNA, by sequencing those DNA and RNA molecules, or even sequencing the proteins present within a biological system. So once this data we generate by wet lab experiment. Wet lab experiment means the research that we do it in the laboratory by taking some biological sample. So once the data are generated, then we apply informatics techniques for analysis. This is very important. Simply, if you generate some biological data and if you cannot handle and cannot say anything about those data, then those data are of no use. So those data we analyze it by using the informatics techniques, where we use the mathematical, computer science, and statistical formulas, and then finally it gives rise to good information for us. So let us come to the aspect of biological data. As I said, in the laboratory experiment, by extracting the entire DNA of a bacteria or for a virus, and then we can sequence it and find out the nucleotide sequence of the DNA and RNA molecules. And once we get those sequence data, then these are usually very complex in nature. The data are very huge data, which manually we cannot go for any interpretation. So biological data refer to all the wet lab experiment where we can generate the data. So one of the characteristics of biological data are heterogeneity. That means a lot of heterogeneous. The simple example is that when we say the SARS-2 virus, so this is not only one type of virus is circulating. Now, in due time, when the virus mutations occur, then the genetic data, some changes occur, and that gives rise to some strains, what we call as the UK strains or some Brazilian strain, etc. So these are heterogeneous. Not only the single kind of virus, but the related viruses. We had a different type of the coronaviruses in animals as well as in human. Now, if you see them as a single virus, then it looks like a same kind of virus. But when we go for analysis of its genomes, then you'll find they are heterogeneous, not homogeneous. Then complexity, because this data they give rise to different kind of molecules, a complex nature of the data, where it is very, very difficult to go for interpretation because of this complexity. And then variability. These are the nature of characteristics of biological data. There are a lot of variations occur within the same kind of organisms or same source of data. So this data, it is not possible to handle manually. So we need their computer-assisted software to handle these databases, and within a very short period, short span of time, sometimes even in seconds, this software can analyze and give you some very valuable information about the biological system. So this bioinformatics, it involves the wet lab experiment, where we can extract the genomes and the nucleotides, even the proteins, proteomics, we can do it. And then we can generate the data, and those data we can put it to some software, and the softwares will analyze and give us the information. So this bioinformatics also involves the maintaining and design of software and databases. Until and unless we have the databases, we cannot go for any kind of interpretation. This is one of the most fascinating part of the bioinformatics subjects, where the databases, global databases are available, and most of the databases are open access. Anybody can access and get the information for interpretation of your results. Okay, so this involves not only the software but also databases. So certain supercomputers, this enormous amount of biological data contributed by the people throughout the globes are stored in a very systematic order so that it can be used for analysis of data. Okay, so even from our institute, we have submitted thousands of genetic data to these databases and can be retrieved at any time for analysis purpose. So if you go through some broader areas of bioinformatics, then you'll find mainly the genomics, proteomics, drug designing, computational evolutionary biology. This is extensively used to find out the relatedness about the organisms, how much they are related to each other, can be find it out. Measuring biodiversity in a particular area, what is the intensity of diversity among the biological system can be measured using this bioinformatics tool. This is another area some people are constantly working on to find out the biodiversity by using this science of bioinformatics. Then not only that, the modeling biological system, it is now very much possible, some sort of a virtual kind of a molecule you can design or you can model it. That's again somehow related to drug designing. So we can, taking this information, so we can find out and design some drugs in the virtual platform. But at the end, definitely, we need to perform the wet lab experiment to find out the drugs that can be used for treatment purposes. For example, by using this bioinformatics tools, we can propose to develop some viral vaccines. So once all area can be explored, then we can actually apply it to the wet lab experiment for designing vaccines. So in the previous classes, all of you have already learned about the polymerase chain reactions, where a specific part of the genomes or the DNA we can amplify for diagnosis and detection purposes. So using this bioinformatics tools, we can design the primers. So until and unless we have the information about the genome in the database, we cannot design a primer for diagnosis purpose. So designing of the primer for diagnostic purpose, we use the bioinformatics tools for, and we use those databases to extract the genetic informations, and we can design the primer, and it can be used for diagnosis purpose. So functional and structural genomics, where in the functional we can go for analysis of gene expression. If we have the nucleotide sequence of a particular organisms, we can find it out how many probable gene will be there. And these genes are controlled by which are the sequences. So all these analysis we can do it by using some computer-assisted models. So analysis of regulation, gene regulation, the analysis of mutation in cancer, like where the mutations occur and because of which particular area the mutations leads to uncontrolled proliferation of cells, or what we call it as a cancer, can be detected by using bioinformatics tools. So this field has been extensively used in different areas for quick and accurate interpretation of data. Before the science was available, people were not much knowing about why actually these things are happening, where actually the mutations are occurring, which are the area of the genome where they are vulnerable for mutations, were not known to the people. But now anyone can explore and can find out using the bioinformatics tools. So similarly, the structural genomics, where the sequence analysis, genome annotations, then comparative genomics, we can analyze it by the bioinformatics tools. Okay, so if I give you some set of nucleotide sequences, so we can't go for any interpretation which organism this particular sequence is without using this tool, but by simply using some of the open access softwares, within few seconds you can interpret this particular sequences of which organisms and which particular gene, and it coded for which protein. All information anyone can access it. I will try to give you an demonstration towards the end of this lectures. Okay, so similarly, not only the genomics, it is also related to proteomics, where we can go for analysis of protein expression, prediction of protein structures. This is quite interesting, and nowadays, suppose should I go for this one? Yeah, like this way. This is the coronavirus. Commonly everywhere we see the pictures, and you can see some of the spike proteins on the envelope of these viruses. Now, why this particular they have drawn it like this way? Why not a single different shape? It is because of this is the trimeric spike protein structures, and this is a model that is generated by using bioinformatics tool. This is what we call as the molecular docking experiment. Simply, we need the nucleotide sequence of that particular gene. Suppose this is a particular nucleotide sequence that you have generated by PCR and sequencing in your laboratory, and using this data. This data is very vague for us. We cannot say anything about this particular data, but using this data, when you put it in certain software, it will give rise to the protein structures, how they look like, and what are the different areas, which areas of the proteins are responsible for binding with the cell surface, and which remain anchored to the plasma membrane of the viruses, the cells, infected cells, can be analyzed. So this predictions of protein structure is a very interesting field, where simply getting a sequence of the organisms, we can go for prediction of protein structure. Now, most of the time, these predictions can be prepared and generated in the laboratory, and finally that give rise to active biological molecules, can be used as a drugs or can be used for diagnostic reagents. So all these are very well now possible using the bioinformatics tools, and for that limited facilities are needed. Only thing is that we need to know the science of the bioinformatics. Now, the biological databases that we needed in bioinformatics are mostly the sequence, structure, and literature. This is quite interesting. So literature is also a biological database that we essentially we need it for analyzing our data. So the bioinformatic databases provide information of any kind of sequence, structure, as well as literature. So just I'm putting some of the amino acid sequences of certain proteins, and this is the nucleotide sequence of some viruses. So as such, if we see, we cannot tell anything about which organisms or what gene this particular region is, but simply today itself we can just copy it and paste it and go for an analysis. Within few minutes you can say this is related to which organism. So that is the way how the bioinformatics help us in interpretation of data. So some of the things that very frequently you heard about, that the double mutant of SARS-2, DC614 and Z614, this that straightway means that there are some mutations which we could see at certain points where in the spike protein in the position 614, certain strains, they are aspartic acids, and some are the glycines. So these are because of the mutation that occur in certain particular triplet codon of the nucleotides, and that give rise to different strain. And now scientists could establish that because of this mutation, the viruses are more adapted to the human cells and readily can get infected to a human person. So this is in the process of virus evolutions, they get adapted in such a way that certain mutations, they induce it, and then they are more adapted to the particular system. So basically, as you know, this is a bat coronavirus, which later on has been shifted to the human, and now it become a human virus. So this whole process needs adaptation of the virus. Now, the general way we can say this is an adaptation, but at molecular level, where actually the changes occur, we can explain by using the bioinformatics tool. By analysis of this genome, using certain software, exactly we can say that, yes, this 614 is the point where the mutations are observed among the coronaviruses. So that is the initial study only, but not only one position. There will be many other parts of the genome where the changes are occurring on a regular basis, and that give rise to some new strains or new types of viruses. So within a very short span of time, how people could able to explain it with such an accuracy, a single amino acid changes taking place among the viruses, is possible only by utilizing the bioinformatic tools. So this is the science of bioinformatics, which has really revolutionized the whole concept, and it become very, very fast for us to give some interpretation and explanation. So this is an spike protein. As you know, this is the most immunogenic part of the viruses, because against which we are taking the vaccines. So these spike proteins, some genetic information is there in the RNA molecules of the virus. So that part has been sequenced, and all the sequence informations are available in open access databases. So from the databases, we can take the sequence and we can go for modeling of the protein. This is one such model people has prepared already, and they found that these spike proteins look like somewhat of this shape, where it is having a transmembrane domain, and then they are having fusion proteins and terminal domain, then binding with the ACE, angiotensin receptors in the human cells. This is the part which is responsible for binding with the host cell. So with such an accuracy, now it is possible to explain the structure of viruses. So without this kind of protein modeling and bioinformatics tool, it is not possible for us to go for interpretation up to such an extent. So these are the different databases, like primary sequence database, protein sequence database, protein classification, pathway, microarray, 2D phase database, public literature. All these are biological information generated in different laboratory. Even like from all laboratory, our institute also, we generated some sequence data and we submitted to those databases, and they are maintaining those databases, and anybody can explore these databases. Now coming to the third, where actually you will get it. These points are important for you. Every one of you must remember. This is something very remarkable things has been occurred in the mankind, like in the entire globe, and in the 3 continents. One is this European Bioinformatics Institute, this just near the Cambridge in the UK. It's called as the EMBL. These are actually some laboratory who are maintaining some supercomputers and use data they can store in those supercomputers, and they are linked with some other databases. The second database is the GenBank. GenBank is housed in NCBI in New York, the National Center for Biotechnology Information, maintained by National Institute of Health, USA. It's called as the GenBank. This is another area where they maintain a lot of supercomputers which are storing an enormous amount of data contributed by the scientists throughout the globes in a very systematic manner. And the most interesting thing is that anybody can access this data. There is no restrictions for anybody. And third is again located in Tokyo, Japan. It is the DNA Data Bank of Japan, DDBJ. So these are the three databases where they maintain the nucleotides sequence databases, they maintain the protein structure databases, they maintain all the other information as it has been said, the pathway, microarray, 2D base, all, including literature. This is quite interesting. These platforms also maintain the literature. They all link up with the different journals, and from them we can access those journals. At the same time, the data, when we extract it, we get a link to get access to those journals, and we can study their research work, everything. And all these three supercomputers are interlinked and through some common platform, either EMBL or GenBank or DDBJ websites, we can access the data present in those areas. This is a huge and enormous data, including all creatures in this world, starting from microbes to an elephant to whatnot, all plants, animals, or viruses, bacteriophages. For all these organisms, you will get the genetic informations. These are the pooled data maintained in a very systematic manner, and it can be accessed by anybody. So EMBL, GenBank, and DDBJ, we say that in 3 continents, this labs are present and they're interlinked to the net, and we can get access. Simply typing the NCBI, the Google page, we can get to the GenBank data, where we can retrieve all the protein information, genome information, structures, taxonomy, books, nucleotides, everything, so you can access it. So this become very easier for the people, for the scientists to study these organisms. Before development of this kind of databases, there was some restricted or limitations were there among the people to study these organisms, to find out the relatedness at the pace how we could able to say about the new strains or the new type of viruses. It is only possible because of this kind of databases accessing to the people. So earlier, the people who are not able to get those informations, and mostly the informations were mostly these informations were limited to the scientific journals or the textbooks. So rarely people used to get the full access of this data, and the pace of research was also less. Now it becomes easier, and anybody can access and go for interpretations out of their own. Even like some organism which are not at all present in our area, we can study those organisms by retrieving the genetic information from these databases, and we can go for predicting proteins, we can go for predicting modeling some new drugs or any molecules by using those databases. This is the DDBJ. Anyone can access it. So I'll be just winding up, and finally I'll come to a point like at NCBI, they are maintaining a particular program called as Basic Local Alignment Search Tool. I'll request all of you to note it down, the full form of BLAST. It's called as Basic Local Alignment Search Tool. This is an open access software. I'm again requesting all of you, please note it down. BLAST, Basic Local Alignment Search Tool. This is the very first tool that we use it when we get some sequence data of an organism to find out what kind of organism and what kind of sequence it is. So for that, the NCBI, they have launched an open access software called as BLAST. So this BLAST program, when we fit our informations and request the software to analyze what kind of genetic information it is. So it will go through the genetic informations that's stored in all these supercomputers at EMBL, GeneBank, DDBJ. Together it will search it, your data. They will try to compare with where it is matching. And finally, within few limited periods, 1 minute or less than even 1 minute, it will give you the 100 best results where it is matching. So once you get the matching, you can find out the organisms, and after that, further we can analyze it, what kind of gene it is, and which protein it is responsible for, whether it is a protein coding or non-protein coded reasons. This can be analyzed. So when you get some time, I'll ask you to try it at your level, just for your interest. Even like if you don't have any sequence information with you, simply in the search engine, you can type a virus name and you can get the genome informations, and you can open it and can see what is the total length of the genome and what are the different genes located. So this data can be accessed by anyone by using the software. This is a part of GeneBank. Well, here I should say this, you should not confuse with another word called as the GeneBank. In your syllabi, there is a topic called as the GeneBank. I guess has been discussed separately. GeneBank is related to the maintaining the genetic pool of all creatures in the world. They are maintaining the genetic. But this GeneBank is different. GeneBank is a collections of nucleotide sequence as well as the protein sequence in that virtual mode or in the computers. This information. This is not related to actual the creatures or any sperm or worms preservation. So GeneBank preserve all those genetic informations in actual biological form, but in the GeneBank, it is the genetic information in the form of nucleotide and the protein structures and sequence informations are maintained in the supercomputers. So basically, we use this platform for analysis purpose. Not only that, once you get the information about your nucleotide sequence, then there are certain open access softwares available in the internet. One such is called as Mega software. It's a molecular evolutionary genetic analysis software. This is another open access software. We can download it, but we need to know how to use this particular software. So using this particular software, we can find out the phylogenetic analysis of your organism. For example, how much there are the viruses, the example of SARS-2 virus of India is related to that of China or South Africa or Brazil, where the changes occur, whether they are of same type or there are some differences that can be analyzed by generating a phylogenetic tree. More particularly, this molecular evolutionary genetic analysis tools are extensively used to develop some kind of a tree called as the phylogenetic tree. It's look like a tree, just the branches of a tree, but these are prepared not simply by drawing with scale and pencil. These are software generated, and the length of this branches are having some importance. The structures, like from a particular point, this two group of microorganisms get diversified in the evolutionary process. Basically, they are of the same kind, but at certain point of evolutions, even we can predict at what particular point, how many millions years before these things has taken place. All are based on certain statistical model. So this kind of trees are designated as phylogenetic tree. This is another outcome of the bioinformatics tool through which we can generate the phylogenetic tree. Now, as you understand about the bacteria, archaea, eukarya, so how they are diverse with each other. They had a common ancestor, but at certain points they get diversified. The bacteria, they have branched separately, whereas the archaea, they move to certain part, and at another points, which is far after this particular diversification, they are diversified to archaea and eukarya. So again, the eukarya, different branching, the organisms are diversified, and finally it gives rise to the animals, fungi, plant, like that way. So this kind of, these are all based on the nucleotide sequence analysis by generating the phylogenetic tree. On the basis of how this nucleotide get changed in the evolutionary process, we can generate this kind of phylogenetic tree, which clearly indicates that two organisms, how much they are related to each other. So all these different, as you can see in the eukarya, they are closely related and are diversified from archaea. And finally, all of these two are much more diversified from the bacteria. So even again, among the bacteria, we can see the diversification, how much they are closely or distantly related, like gram-positive and like spirochetes and cyanobacteria, where they are diversified at what point of evolution. This is what we call as the evolutionary genetics. And we are having some software. One such software is called as MEGA software, Molecular Evolutionary Genetic Analysis. This is some inputs I try to give you, apprise you about this area of bioinformatics. Maybe some of you may get some more interest to study about it, and this itself become a separate discipline in the biology, where the bioinformatics are extensively used. Now, look at these particular pictures where leading to the present context, we know about the SARS and the palm, through the palm civet in 2002. The SARS infections from this bat has come to the human, and at that time, around 700 to 900 people die, but finally it doesn't just spread it to other area. This is the first incidence of SARS. We now call it the SARS-1 infection. And from bat, it was an established chain how from bat to the civet adaptation takes place, and finally it has come to the human. But somewhere again from some other kind of bats, again the MERS, the Middle East coronaviruses, which affect the camels and can also affect the human. These are some of the episodes that earlier occurred. Then in between, there were the evolvement of Nipah viruses through some fruit bat. It has adapted to the swine, and then it has finally disseminated to the human. Now, this kind of establishment, like how it has come, is only possible by using certain bioinformatics tools to correlate the viruses, how it has adopted and disseminated to the human. And finally, in the present episodes, these are some questionable things. SARS-CoV-2 intermediate is still unknown, which will be there. Some people say about this pangolins, maybe one, but maybe some say the civet, or there is some questions mark. Is it directly from the bat to the human or without any intermediate host? These are some of the areas people are now focusing to find out how it has occurred, this one. Similarly, the Ebola viruses, without any known intermediate host from fruit bats to the human, transmitted in the Congo in Africa, and later on it become human-to-human transmissions and become a very dreaded disease for mankind. So this kind of this is another, probably this is the last slides I'm sharing with you. This is a comprehensive phylogenetic trees which are generated on the basis of the nucleotide sequence of the coronaviruses from different sources, including the earlier MERS and the other coronaviruses of cattle, dog, horse, as well as the human coronaviruses, and to the present context of SARS-CoV-2. So it could, from this kind of phylogenetic tree, this is very clear that the present SARS coronavirus 2 is much more related to the bat SARS like COVID sequences in the phylogenetic analysis. So although they are having some near relatives of bats and the humans, earlier incidents of say 2002, but are distant from other coronaviruses, which are grouped under different genus like Hibeko, Nobeko, and Meko. These are different group of viruses. So the phylogenetic analysis could give us much more accurate information about the genetic relatedness among the organism on the basis of each nucleotide sequence pattern study. So this whole science is related to the bioinformatics tools. So nobody can just generate this kind of pictures until and unless we use some computer assisted models for generating this relatedness. So like this way we use the bioinformatics tool. This is a very simple explanations about the science, but this is only 1 hour class, so some basic idea I try to focus on, highlight on you. Could you see my slide now, Google slide, Google page?
Yes, sir. Yes, sir.
Okay, I will just show you if you had a sequence like suppose I'll just copy this particular sequence, nucleotide sequence that you have generated in your laboratory from an PCR and sequencing. And then what actually this particular sequence is about. Then we can simply just type NCBI in our Google page, and then it will give rise to the, it'll take you to the National Center for Biotechnology Information, NCBI. This is an, as I said, that this is a laboratory located at New York, and they are maintaining this supercomputers having the enormous data. So we can go through the first page of the NCBI, where you will get a program called as BLAST. Here, my cursor is coming here. If you just click it, BLAST, so it will, of course, we need a very first internet for this kind of using this software. We can see here different kind of the BLAST program, BLASTX, BLAST10, Protein BLAST, and Nucleotide BLAST. So since our sequence is a very short one, so I'll select this Nucleotide BLAST. If you just, any one of you can do it. So in this particular enter query sequence, the sequence. I have copied it from the page. I'll simply paste it, Control V. I'll just pasting this sequence. I don't know what is this particular sequence. I will request this software to compare with all kind of databases available in their supercomputers for showing the highly similar sequence by performing a software called as the MegaBlast. So I will ask them to show results in the new window, and I will request for basic local alignment search tool to analyze this. Now it will take few minutes or maybe a few second time. It's all depend on the internet connection. So it is showing that. Could you able to see my slide?
Yes, sir.
Yes, sir.
Fine. See, within few seconds, it has given me the information. What is telling? See, it is a sequence of foot and mouth disease virus type O. Did you see? They are giving the 100 best results, and all are foot and mouth disease virus. So this is an online software. I have used it from raw data like this particular sequence data. I copied it. I put it in the BLAST software to find it out what actually this is related to. The software is saying this is foot and mouth disease virus. It is related to FMD virus. Okay, and it is showing some person identity up to 100% with other sequences. And this is one SSN number. If I click this SSN number, then it will give rise to the gene bank database. This is the gene bank database. Can you see it is the gene bank database. It is having a title, foot and mouth disease virus, type O isolate from IND, probably it's from India. Yeah, it's from India. And if you scroll it, then you will go to the details about it. Yeah, this is the whole genome sequence about the foot and mouth disease virus. It's from India, Meerut, Uttar Pradesh. They have isolated in 2009. Did you see? And then the whole information. This is the polyproteins, and then these are the different map peptides, and these are the sequences. So this particular data, very quickly, we can download it from this particular websites to your computer for analysis purpose. Otherwise, this is one aspect. So even what we can do, if you don't have any sequence with you, for example, from the whole here, we'll just select these nucleotides we are interested. And here, let us type SARS-2 virus. Simply you type it, SARS-2 virus. Yeah, it is showing the database severe acute respiratory syndrome coronavirus 2 reference genome. So the entire genome structures, this having the open reading frame, and these are the different coding regions. If you wish, you can download it, or we can see this genome. This is the gene bank. This is online. Means it's a Wuhan 1 type complete genome. When in 2020, 18 July, they have submitted the coronavirus. The genome length is 29,903, almost in and around. Some of them they may have a little bit more or less, but it is an LERS virus genome, 29,903 base pair. They have published it in the Nature journal, so we can go through the publications. Then what else? If you scroll it, then gradually we can come to know about the it's from Homo sapiens, Wuhan, in the December 2019. Probably this is the first data that we are having in this corona episode. And when you scroll it gradually, you will get this other protein informations. And then one by one, ORF1, ORFB, this is NSP2. These are different proteins, and somewhere you'll get the spike proteins against which the vaccine has been prepared. So we can get this, and these are means openness to anybody. Anybody can derive and retrieve this data from the gene bank. So these are the part of the bioinformatics, physical information using the computer software and databases. We can go for interpretation. So we can download the different source to coronavirus from different countries, and then we can see how much they are related to each other, where the mutation occur, what about the double mutations, why it's called, and where the actual position. So all this analysis we can do it by using this kind of software. And this is the subject what we call as the bioinformatics. Of course, this is not the end. This is a very preliminary idea I'm giving to you. But as you know, this become a trend. Anybody can ask you how we can say that this is a different strain and this is a different strain and the variations. Then you can explain it that using this bioinformatics tool, we can find out the mutations occur in the different type of virus, which give rise to the different strain of coronaviruses. So just an example, I have taken it here for SARS-CoV-2, but for all the viral infections, we can study and generate the data and can analyze it. So it becomes quite easier for us by understanding this subject of bioinformatics. We can analyze and interpret the data in an accurate form. Okay, this is just a brief introductions to the subject bioinformatics. So thank you very much for this particular sessions. But again, at 2 o'clock, we'll again start the general biology course. Yeah, thank you very much. If you have any special query or things, you can just post me.